Good afternoon. It is great to be able to be with you, even if it's just via this video. I would have loved to have been able to come to see you and be in the seminar today, but unfortunately it was not possible. But today we're going to speak to you about abortive disease and in particular IDVet solutions for the diagnosis of abortive disease in ruminants. So I am Anna Greatrex. I am the export manager here at IDVet, and I'm here with...
I'm Liz Gravis, and I manage and develop PCR line with IDVet.
So this, I think Mike has already spoken to you this morning about IDVet genetics and who we are and what we do. You've heard from Linda about abortion disease in general, so now we're just going to go into some more detail about IDVet products for these diseases. So Mike, you can change the slide. So we're just going to start with a quick introduction about abortive disease and our solutions. So as you know, abortions cause huge economic losses to producers. It is often very difficult to detect the actual cause of the abortion, but we can try by using both direct and indirect diagnostic methods. Pathogens which can cause abortions are numerous. We have Coxiella burnetii, Chlamydia, Brucella, BVD, BTV. So IDVet offers diagnostic solutions for many of these diseases. Now, in order for the pathogen to be able to provoke an abortion, it must cross the placental barrier. And if this is the case, the pathogen can either directly cause death of the fetus or can indirectly cause death of the fetus by causing lesions in the placental membrane. Also, many pathogens can provoke fetal malformations, cerebral lesions, so the effects of pathogens on the fetus are quite variable. So this slide now, Mike, is a slide about direct detection, PCR. So PCR has the great advantage of being able to establish a direct link between the abortion and the pathogen by directly detecting the pathogen either on the fetus or in by taking a cervical swab or a vaginal swab from the mother. Liz will go into more detail about the different matrices, but of course PCR is one of the major techniques we will use to try to understand what caused the abortion. Serology may also be used to better understand what could be going on in the herd. IDVet, we've specialized in ELISA kits for many years. So serology is of course an indirect detection method. The fetus will only become infected if its mother has become infected, so we can look at the immune status of the mother to get a better picture of what's going on. But the immune response that we detect may be due to a much older infection, and so a positive serological result does not necessarily establish a direct link between the pathogen and the abortion. We can, however, use serology to rule out a certain number of pathogens. So a negative result for a certain pathogen at least lets us know that this pathogen was not the cause of the abortion that we're looking at. So we can look at negative serological results to better understand what's going on. We can also use serology to determine if there's a seroconversion between the moment that the abortion takes place and several weeks later. So we can look at an increase in antibody levels after the abortion, which can also implicate the pathogen in the abortion event. And in that case, we'll be testing serum from the mother. So the next slide is a comparison between ELISA and PCR. So we have indirect diagnosis on the left, direct diagnosis on the right. So as I've already said, a positive serological result from the mother cannot establish a direct link between the abortion and the pathogen. But if it is negative, we can rule out that pathogen. And for a number of diseases, we're going to have to test twice to have a double serological exam at the time of abortion and 2 to 3 weeks later, because at the time of abortion the mother may not have had time to seroconvert and have a immune response. So it's very important to do a double serological testing for certain pathogens that we have listed here, for example, Q fever, IPR, Salmonella. On the other hand, if it's positive PCR, it's very likely that the pathogen is implicated in the abortion. And if it's negative, once again, we can exclude that target from our diagnosis of the abortive event. So now we'll move on to the slide about the IDVet test for abortive disease. So Mike, you can skip the slide just before. So the next two slides show the diseases for which IDVet can offer you diagnostic kits, either ELISA or PCR. You can see that for Acaban we offer competitive ELISA. For blue tongue we have both ELISA and PCR, and so on. We will go into more detail about these diseases during the presentation. So you can see the diseases for viral diseases, and on the next slide you can see the diseases for bacterial disease and for some parasitic diseases. Okay, so we'll go into more detail right now about these diseases. So I'll let Lise explain to you how we've gone about.
Okay, so you understand that diagnosis on abortion is very complex, and a lot of time we don't find why the animals have an abortion. So our proposition and our strategy is both. First is to propose a screening direct in PCR with a system on one extraction. I explained just after. And the second size is to propose combine PCR and ELISA to find the good combination to have the response, the answers about the question why the animal have an abortion.
So the next slide.
The IDVet PCR kit for abortive disease. So first, what is available today? We have first Anaplasma phagocytophila, pathogen come from ticks, very high prevalence in France. I explain after. We have also chlamydia, SPP, Q fever, toxoplasma, SPP, and neospora. Very soon we have available Listeria monocytogenes, Salmonella SPP, BHV4, and Brucella. The strategy we propose is you have an abortion, and all countries have another male pathogen. You choose your pathogen, 1, 2, 3, and we put in the same boxes 1, 2, 3, 4 pathogens to have a pack abortion in your country. It's very flexible. So you choose the pathogen, and more you have pathogen, more the price decrease for the pack. It's the first point. So you win a little bit in screening a lot of pathogen. Second size, for the extraction. I explain after for all pathogen what is mainly matrices for extraction. With our extraction, we propose one extraction for all the pathogen is the same with spin colon or in magnetic beads. Exactly the same. So, for example, if you choose a matrices, for example, swab, you make one extraction and you can amplify with a lot of amplification. So you win also with the choice of matrices. So that is the first point. You can have an economic system with extraction, and a second with the pack system. More you put pathogen, more the price of the point decrease.
Okay. So the next slide, we're going to now look at each individual disease. We're going to start with Q fever. So Q fever this is data from Belgium. In Belgium, for example, 50% of herds are infected have been exposed to Q fever, but Q fever is responsible for about 2% of abortions. In any case, authorities always look at Q fever as being a potential cause of abortion. You can use a combination of serological and molecular methods to try and elucidate what's going on in the herd. So here's an example where bulk milk is tested by ELISA and by PCR, and depending on the status by the different techniques, you can come to a certain conclusion. So for example, if you have a positive result in bulk milk by ELISA and by PCR, you can say, well, the herd is infected and there's active circulation of Coxiella. If it is negative by serology but positive by PCR, you can say the herd has been recently exposed to Coxiella, but maybe seroconversion has not yet started. So in this case, you're going to want to test again several weeks later to see if animals have seroconverted. If you have positive ELISA, negative PCR, you're going to come to the conclusion that this was an older infection. So animals have antibodies from an older infection, but Coxiella is no longer circulating. And if both techniques give negative results, you can say that the herd has not been exposed to Coxiella and that there's of course, no circulation going on. So this is an example of how ELISA and PCR are very complementary techniques to try to understand what's going on in your herd. So the next slide is about the IDVet's indirect ELISA for Q fever. So many of our indirect kits use the multi-species conjugate, which allows us to detect antibodies in multiple species. This conjugate works with mammalian IgGs. So we've had certain researchers that have studied Q fever in rodents with our kit and in many other diseases. Of course, the primary use of this kit is in ruminants. This kit, of course, can be used to test serum from the mother at the moment of abortion and a few weeks later to see if there is seroconversion has occurred, and in that sense you can try to understand if Q fever is implicated in this abortion, but you're also going to want to have information from molecular methods. So Lise will speak to you about the PCR for Q fever.
Yes, about the PCR. So for this kit, we have a triplex. To be beginning, the matrix, the main matrix used on Q fever is a swab on placenta, cervical, and vaginal. We can also use organ, spleen, placenta, or tissue. It's possible to find also in feces, more for epidemiologic information, and the best matrix is gastric juice. This kit have a very good limit of detection because it's less than a copy by PCR. All our kits are validated on this limit of detection. Is mandatory for us, is important. The specificity is validated on a panel come from OIE laboratory in France, and the specificity is 100%. The triplex, what is the triplex? The first detection is of course for Q fever in farm dye. The second target is internal control endogenous to check if in your matrix you have cells. For example, in swab, you don't see cells in the swab to compare to blood or organ. So to be sure you have cells on the swab, it's important to have internal control endogenous. In second point, we have also exogenous control. Our exogenous control is bacteria. The quantity is very low, and it's another bacteria different to Q fever. To be sure that in all the extraction, all step is validated. We weight a quantity, a CT value on this internal control exogenous, and to be sure that if the CT value is correct, you have not inhibition and you have not problem of extraction. Because for example, on milk or on feces, on organ, it's very inhibition matrices. So second point, on the control, we have also a positive control. This positive control is not a plasmid. The plasmid just validates if the probe and it's correct and the system is correct. But we have more that. We have a real sample, supernatant of swab, and inside we have bacteria, dead bacteria, come from the reference lab in France. And if your control is positive, you know that all the manipulation or the handling is perfect, okay? And this control is very low. CT value is about 32. So if you have this CT, 3 CT before and 6 CT after, all manipulation is correct, and that is for all our kits. And of course, if your fetus or placenta or sample is positive, in abortion case, it's very probable that the abortion is due to Q fever.
Chlamydia abortus. At IDVet we have a multi-species indirect ELISA. It's based on a peptide antigen, the major outer membrane protein, protein 90, P90. This protein is highly specific to Chlamydia abortus and also very immunogenic, which means that the kit has high sensitivity and also very high specificity. We use a peptide, whereas other commercializers and even the complement fixation test will be based on whole bacteria or recombinant proteins, and thus will have more cross reactions with other chlamydia species. So the kit is specific to abortus, very sensitive, and once again you can use it in the context of abortions to see if there's seroconversion between the time of the abortion and several weeks later. If there is seroconversion, you can conclude that chlamydia could be involved, a cause of the abortion, but you can't make a direct link between chlamydia and the abortion. If the result is negative, well then at least you can say that chlamydia is most likely not the cause of this abortion. But to go further, you should do a PCR test.
So for the PCR, it's a duplex, it's chlamydia SPP, and for this kit, the internal control is endogenous to control if presence of cells or not. For the matrix, you can use swab or organ, okay? And the limit of detection is very low, and the specificity is evaluated by the reference lab in France in a reference lab panel. It's 100%. In the positive control, it's like the other kit, supernatant of swab with doping with bacteria inside, very low to control all the steps. If you found a positive in a chlamydia, in a fetus, or in a swab, or in organ, the abortion is probably due to the chlamydia SPP.
Now moving on to Neospora, once again we have an indirect multi-species ELISA for Neospora. It will work both on canine sera and ruminant sera. The test for Neospora is a bit different than the other pathogens that we've discussed, because for Neospora there's a very, very high correlation between a seropositive result in the mother and the probability that the fetus is PCR positive. So in that sense, it is possible to test the serum from the mother and to conclude that Neospora is most likely the cause of the abortion. So in this case, you can have, it is not necessarily important to do a PCR test, but if you want to go further, you can.
Yes. If you want to be sure that the abortion is due to Neospora, you can find Neospora in the brain or in the heart muscle. And also you can use, but it's not a good matrices, in swab or on the cervical, and swab or the placenta. But it's not a lot of mycobacteria, and you find very low positive. But if we have not other matrices, you can use it. This kit is with an endogenous internal control. The sensitivity is very good, and the specificity is 100%. We have evaluated the specificity with an expert lab in France, not with a reference lab because it's not available. And we make sequencing of all the positive to be sure that it's positive. So this panel is very good, and we have 100% of specificity. For the control positive, the positive control, sorry, it's a brain doping with the bacteria. And of course, if you have positive in PCR, the aberration is due to Neospora. We have also developed a triplex with Q fever and chlamydia with internal control endogenous. Why we developed that? Because in France and in a lot of countries, for example in Italy or in Belgium, it's the mainly pathogen present. So for economical diagnosis, we develop a triplex. So it's exactly the same matrices you found in the duplex system in chlamydia and triplex in Q fever, but here you have 3 detection, one for Q fever, one for chlamydia, and one for internal control.
So moving on, for toxoplasmosis, we also have a multi-species indirect ELISA, which can be used for seroprevalence studies, or also to determine, to exclude, especially to exclude toxoplasmosis as a potential cause of the abortion. It is once again a multi-species ELISA based on the P30 antigen.
For the PCR kit, we have developed a duplex with internal control endogenous. The mainly matrices is brain, placenta, and earth musk, like Neospora. And the sensitivity is very low, very good, and the specificity is 100%. We evaluate the specificity with the reference lab in France, is the same in chlamydia and in toxoplasma. For the control positive, it's a supernatant of placenta doping with toxoplasma. And if you have positive in toxoplasma, in PCR, you know that abortion is due to toxoplasma.
Now, for Schmallenberg, we offer a range of ELISAs, indirect, competitive, and milk ELISAs. The reason for this is that when Schmallenberg first appeared in Europe in 2011, IDVet was the first company to produce a diagnostic test for this disease, an antibody test for this disease. The first test which was used was virus neutralization, which is, as you know, very difficult to implement on a wide-scale basis. So we launched the first ELISA on the market, and only 3 months after the virus was first identified in Germany. The competitive ELISA is an optimized version of the test with improved sensitivity for sero with low VNT titers and and earlier detection of seroconversion. The milk ELISA is being used very effectively on the periphery of the outbreak to identify new, to follow the spread of the virus to new regions. So milk is a very cost-effective method of following the spread of a new outbreak. Now with Schmallenberg, what's very interesting is that the viremic period only lasts 2 days. So of course serology is quite useful to identify infected animals. But you can also use PCR if you're lucky enough to be in the viremic period.
For PCR, Anna explains that the viremi is very short, 2 days. So we can find in PCR on the mother. What is interesting is to test if the mother is pregnant, for example. In the fetus, you can find the virus in brain, and also you can find on the swab in the placenta. But this matrix is not very high sensitivity. We can find, but the CT value is very low. This kit has a very good sensitivity, and the specificity is validated by the reference lab in France, and also with the panel of strain come from FLE in Germany. The positive control is virus doping in serum, and very low, to be sure, all step is positive. If you are positive in Schmallenberg virus in the brain, on the fetus, you know that the aberration is due to Schmallenberg virus.
Now, blue tongue is also a disease which we've worked on for many years at IDVet. There was a big outbreak of blue tongue. There was a big outbreak of blue tongue in Europe in 2006, between 2006 and 2008. And our kit was widely used during this outbreak, both the competitive ELISA to to detect positive animals, seropositive animals, and the milk ELISA to monitor the spread of the disease to new areas. The kit is based on the VP7 protein, which is highly conserved between serotypes, so the kit will work on all serotypes of the disease. Once again, the serology may be used to, if you have a negative result by serology, you can conclude that blue tongue is not implicated in the cause of the abortion. But on the other hand, if it's seropositive, you should also do a PCR test.
Yes. For the PCR BTV kit, we develop and validate with the reference lab in France also. It's a blue tongue screening detection, so we can detect all the serotype of blue tongue, also the new 27 and 28, so it's worked very well. About the matrices, you can work on whole blood and organ, and we can work on whole blood and convert the mother, and for the positive control is blood doping with the virus, very low, and if the fetus is positive with blue tongue, you are sure that the abortion is due to blue tongue.
For IBR, we offer a range of serological tools, an indirect ELISA, a GB ELISA, which is competitive ELISA, and also a GE competitive ELISA. So the GE ELISA is going to be used in areas where vaccination is performed with a GE deleted vaccine. So if the animal is positive with a GE test, it means that it is a naturally infected animal. The indirect ELISA or the GB ELISA are both used to screen for positive animals in non-vaccinated regions. In France, they use the indirect ELISA to screen. In the United Kingdom, the GB ELISA. It depends on the country. There is some data which indicates that the GB ELISA will have slightly lower specificity, but this is true for all manufacturers and all kits. So the choice really is up to you. And once again, in the case of an abortion, you're going to do a double serological test to see if there's seroconversion. Of course, for IBR, there is no PCR test available. PCR testing is generally not performed for IBR. Moving on to BVD. BVD, as you know, is a very complex disease with existence of persistently infected animals. At IDVet, we offer for antibody testing a competitive ELISA, and then for antigen testing, we offer a PCR kit, which Liz will speak about in a few minutes. In any case, once again for BVD, you will want to do a double serological exam after the abortion, and then on a more of a general level, you can use an antibody ELISA in bulk milk to detect a sudden increase in antibody levels in the herd, which could be linked to the arrival of a PI animal in the herd. So those are two ways that serology can be used. In terms of PCR testing, Liz will speak to you.
For the PCR kit, we develop a triplex. In this triplex, you detect BVTV and BD, border disease 2. In the second dye, we detect internal control endogenous to control if you have cells or not in your sample. For example, in swabs, if you use swab or on serum, because you have not a lot of cells in serum. And also, we have with the third dye a virus, RNA virus, doping, very know about the concentration. If this control move, you have a problem in inhibition or you have a problem of the extraction. So you validate all the steps in all the sample. For abortion particularly, you can use organ, more organ than swab, but you can also use swab. The limit of detection is very low, and the kit is validated with the panel strain come from FLE in Germany and also in a reference lab in France. The control positive, the positive control is blood doping with the virus and inactivated and very low positive. all the extraction on BVD and all pathogen is the same. So if you use this control positive and it's blood, but you use swab in the for testing in BVD, you have exactly the same protocol. So you can validate with this control. So if in your fetus you found positive in BVD, you know that BVDV is probably the cause of abortion.
Now the last disease we'll speak about is brucellosis. As you know, there are many different serological techniques which allow for the diagnosis of brucellosis. These include the serum agglutination test, complement fixation test, the milk ring test, which is not used very widely anymore, fluorescent polarization, which is used a lot in South America. Of course, the drawback is that it requires very special equipment, which is quite expensive. At IDVet for serology, we offer the Rose Bengal test, which is a very cost-effective way to test samples. The drawback being that it's a visual test, and it's a bit more difficult to implement for a large number of samples. We offer, of course, the indirect ELISA, which is very good if you have a lot of samples to test, and it is probably the most sensitive technique among the different techniques for detecting antibodies for brucellosis. And a word about the ELISA kits we offer for brucellosis. All Rose Bengal diagnostics for serology are standardized according to the OIE standards. So in our laboratory, before we release any batches, we must test them with OIE standards to ensure the optimal analytical sensitivity. And so we have a kit for serum, a kit for milk, and of course the Rose Bengal test.
For PCR kit, this kit is under development. We work hard on developing a specific protocol on milk, but on abortion, it's very easy on the matrices. You can use swab and organ. And this kit is a triplex because it's like the same story like Q fever. On milk and organ, you have a lot of inhibition. To be sure, it's not a problem. You have two internal control. First, endogenous to control the presence of the cells, and the second to control the step extraction and amplification, and if you have inhibition or not. The limit of detection is very good, and the specificity is validated by the reference lab in France, is also the reference lab OIE. For the control, we use vaccine inside, and on the matrix is milk. If you have positive PCR in fetus, you know that your abortion is due to this pathogen, because it's a very mainly important pathogen for abortion disease.
And now just to finish up, we often get questions about the use of our serological tests for brucellosis in the event of vaccination. So there is no DIVA test available for brucellosis testing. That means differentiation between infected and vaccinated animals. Our tests and most commercial tests, if you vaccinate with S19, which is a smooth attenuated strain, you will obtain a positive result in vaccinated animals with our tests. The same is true for the Rev1 vaccine for Brucella melitensis. For the RB51 vaccine, which is based on a rough strain, we should not detect vaccinated animals which are vaccinated with this strain. I do not have specific validation data, but this is what is indicated in the OIE manual. There are, in some countries, there have been, a competitive ELISA has been used in some countries to try to differentiate between vaccinated and infected animals. But this is not a true DIVA test because the competitive ELISA simply has less analytical sensitivity than other tests, and so does not detect vaccinated animals, vaccinal antibodies as well. So this means that the test does not see vaccinal antibodies, it only sees antibodies from highly infected animals, from strongly positive animals. But if the test does not see vaccinal antibodies, it will also not see some naturally infected antibodies, or some antibodies from natural infections. So we would not recommend this type of strategy for DIVA, for brucellosis testing. So basically that is what we wanted to tell you about our range of veterinary diagnostics for abortive diseases. Thank you very much for taking the time to listen to us. We've had a very fun day putting this presentation together because we had a few technical problems, so this is why we were laughing a little bit towards the end. But it's been a very fun exercise to put this video together. We are at your service if you need anything, technical support, any questions, if we can organize any on-site training. Mike is available as well to help you. If you're not using PCR in your laboratory but you would like to, then let us know. We can see how we can help you. And of course, if you need any additional documentation, you can talk to Mike. We have complete validation documents for all the diseases, all the kits that we've talked about in terms of specificity, sensitivity. So all this information is available upon request. And what have I forgotten to say?
I think nothing. It's perfect.
Well, once again, thank you very much. I'm sorry that we can't be with you today, but in a way we've been thinking about you all day, putting this presentation together. So thank you very much, and have a good end to the seminar.
Bye-bye.
Bye.
Thank you.